Okay, a couple of other processes now. Electrophoresis. Electrophoresis is a way to separate fragments of DNA based on their size. And so this process is used for analyzing DNA. How it works, we take DNA, we put DNA onto a bed of agarose, so this is this kind of carbohydrate matrix similar to what is used for plating bacteria. We expose the gel to an electric field, and DNA has a negative charge. It naturally has a negative charge chemically, and so when we connect the environment of the gel to the power source, we're going to create a field in which we have a positive and negative end. And so the DNA is naturally going to move towards, in this case, the right, towards the positively charged anode. And there's going to be a difference between how fast they can move. Because this gel has these little pores in it, the DNA has to physically move through those little pores. And so little pieces of DNA will move more quickly, and thus they'll get further towards the end of the gel, whereas big pieces will move more slowly, and so they're going to stay stuck closer to the where they started. Obviously, in order to get these pieces, what we're using to be able to do that are restriction enzymes. So we're using a single or a blend of restriction enzymes to cut that DNA into those small pieces, and then inserting those pieces into these wells. And what usually happens is that between each of the different wells, we are putting in different samples in which we're trying to compare towards each other. What you can see here is the banding pattern that you see at the end. So each of those little streaks of light, those all represent a particular fragment or sometimes several fragments of DNA. And so you can compare one well to another and to see if there are similarities in terms of where those bands show up. And if the same bands are in the same spot, then that suggests that those original DNA samples were similar. And if the banding patterns are in different places, then that means that that the DNA was very different from each other because the restriction enzymes cut them at different spots. When we do this in class, you'll notice that this picture is a little bit more clear and a little bit easier to see. This uses a type of visualization called ethidium bromide, which is particularly hazardous. It's a known pretty powerful mutanogen, and so for that reason it can cause cancer. And so it's something that is really only going to be used in a pretty sophisticated or a very higher level lab. And so the types of visualizations that we'll use aren't quite as clear, so sometimes it's good to be able to see pictures like this to be able to take a look and see how clear those banding patterns actually can be under better circumstances. The next technique is PCR. PCR stands for the Polymerase Chain reaction, and this is a way to produce many copies of DNA without the use of cells. And so this is an important process, resulted in a Nobel Prize, and it sped up immeasurably the analysis and the manipulation of DNA. So in this case, we start with the target sequence of DNA. So we have some piece that we want to analyze. Primers are going to be created. So these are things that will be created in the lab. Those primers will bind through complementary base pairing with the sequence of interest, generally at either end of the sequence. Then we put the DNA, we put the primers, we put free nucleotides. So this would be remember, nucleotide consists of the phosphate, the deoxyribose sugar, and then one of adenine, guanine, cytosine, or thymine. So this is the raw materials needed to make DNA. Coenzymes that are necessary to allow the Taq polymerase, which is a special polymerase that can work well under these changes in temperatures. It's not going to denature. So we have the polymerase, we have the primers, we have the raw materials, we have anything extra that's needed, along with our original DNA sampler. We put that in the thermocycler, and it's going to regulate the temperature and make copies of that DNA. Thermocyclers come in a variety of sizes, and it just depends on their capacity and how old or new they are. New ones are little tabletop models that can process DNA pretty quickly in a matter of a few hours. So first step, we denature. So the thermocycle warms up to a pretty warm 94 to 98 degrees, and then that's going to cause DNA strands to separate. So that target sequence will have the complementary base pairing, hydrogen bonds will be broken, and it will separate. Next step after denaturation is going to be annealing. So annealing is when, through complementary base pairing, the primers will attach to that target sequence. This is going to happen at a slower, not a slower, but a lower temperature, because otherwise the hydrogen bonding is going to be overcome by that high temperature. So thermocycler cools down at this point. Then the cycle is going to warm up, not as hot as it was originally, but it warms up, and DNA polymerase is then going to move along from the primer down the length of the DNA sequence, add on the free nucleotides that are necessary, and thus we get replication of the sequence. That process will repeat again and again and again. So in application for that, so we saw genetic engineering being used earlier for things like recombinant DNA, using vectors, using restriction enzymes. The idea of PCR is going to be important for genetic testing. So in this case, we're using these tools to look for the presence or absence of particular gene sequence. Restriction enzymes are useful to digest the DNA. PCR comes in to be able to amplify the DNA, because if we have a small sample, it's not enough for us to be able to analyze. We need to amplify it. We need extra copies so that now there's enough DNA that we can use something like gel electrophoresis to actually analyze it. So the most common and easiest to understand application of this would be genetic testing for the purpose of identifying. So we could use this in something like a paternity test, where you're trying to identify whether or not a particular man or woman is the father or mother of a child. And it's also going to be used a lot in terms of DNA fingerprinting. So in the case of, for example, a suspect at a crime scene, if we have a small amount of blood or semen or some type of hair follicle that has DNA in it, we can then we can go through that process of chop up the DNA using the restriction enzyme, amplify the DNA, make the copies through PCR, and then use a gel to be able to see whether or not a particular section of that DNA matches from a suspect to a person in question. So we look at some particular areas. So there are some areas called short tandem repeats where the DNA section is very predictable, and we have restriction enzymes that can isolate these particular short tandem repeats. And these are really useful because they tend to be very different between different people. You wouldn't want to analyze, for example, the results of chopping up the hemoglobin gene, because hemoglobin is something that has very few variations between different people. But some of the non-coding regions do have a lot of differences between different people, particularly once you get out of a family relationship. So, for example, in this particular case, there are a handful of different short tandem repeats, different areas that are being analyzed using a gel. And so there was some semen on the victim from a rape and a murder case. And so we do the gel, and these numbers are representing the places on the gel where we're finding the particular bands. And we can see that the person who was convicted of this particular crime, that in all cases, the places where the bands were different from the places when the DNA evidence was analyzed. And so there was another person who obviously has whose banding pattern matched up with the victim. That shows that it wasn't the person who was convicted, it was this other person who was the one that actually committed the crime. Over the past couple of decades, since PCR and gel electrophoresis have been developed, there are plenty of crimes from the 80s, the 70s, and going on even more recently, as in this one was in the 90s, cases where there wasn't the ability, or there wasn't a reason to do genetic testing, to do that DNA fingerprinting at the time, and so those cases are then being overturned many years later because police departments were keeping some of that evidence, and that DNA evidence was preserved, and then we can still do that testing years later, and to be able to see whether or not the actual physical evidence at the crime matched up with the person we convicted of the murder. Another application of some of these techniques is cloning. And so cloning is just being able to make a copy of a particular genetic sequence. Cloning is a process that happens in not many animals, but it happens a lot in plants, it happens a lot in fungus, in bacteria, and it just means reproducing asexually, so that the offspring is going to have the exact same DNA as the parent. So reproductive cloning, this is one type that you should be familiar with. This is cloning when we are producing an entirely new organism. The process here, if we look at what a plant does, this is pretty simple, and plants will do this very often on their own. If I take fragments of an adult's carrot cell, and I can then shear off individual cells, those cells can divide, grow into shoots, and those little shoots will develop into new plants that could then be genetically identical to the adult from which they came. So that process, gardeners are doing that all the time, because once you have a particular variety of a particular plant that you want, it is beneficial to just be able to make clones of that plant, because you want it to have the same characteristics as its parent, because the parent was already the pinnacle of whatever it is that you wanted for that particular species. Now, as we get from plants into animals, and animals is something that isn't going to happen naturally on its own. And so this is where we need to use a technique, somatic cell nuclear transfer, to be able to accomplish the same result. So we have, on the top left, we have a mammary cell donor. So the donor, as you can see here, is a white-colored sheep, and so we're going to be using its cell's DNA. We take a mammary cell, so this is coming from its udders. We're taking the DNA from that. We're taking the DNA, we're fusing that together from an egg cell. The egg cell came from a black sheep. So the nucleus is removed from that egg so that there's no DNA. It's just an empty egg cell. The DNA is coming from the donor. The DNA goes into this is not a haploid. This was a haploid egg. This is a diploid cell. So that full set of DNA is getting fused into the egg. Now we have an egg that has the full set of DNA in it. We implant that egg with that DNA. We allow it to become an embryo, implant that in a surrogate mother, and then the baby lamb is going to develop and be born. And as you can see, it's going to be genetically identical to its parent. And so the idea of just the white sheep and black sheep here is showing you that the black sheep is not providing any genetic information. It's not the surrogate mother. Also, neither of them are doing anything to influence the end result, which is a sheep that's going to be identical to whichever sheep provided the original DNA. And so this has been done in a variety of different animals. So sheep was one of the first ones that happened. Dolly the sheep was an example there. But it's been done in mice and cats and in many other animals. Not all types is it possible in, and it's actually more difficult. There are conventions against doing this in humans, and even if it wasn't considered unethical or illegal to be able to do so, cloning in humans in this way is more problematic, because comparatively the human egg cell is much smaller, and because it's smaller, it's just physically difficult to be able to manipulate a human egg in the same way that you saw in this diagram with the sheep. Now, what does have a lot of application is therapeutic cloning. So this is not what people usually think of in terms of cloning, but this is the part that actually is going to be much more useful in terms of treating people with particular diseases. What we're trying to do is create stem cells. So we're using cloning to make a type of stem cell called a pluripotent stem cell. Pluripotent means that it has the ability, it's a cell that has the ability to differentiate into any type of cell within the body. So those pluripotent stem cells can come from embryonic stem cells, and so that is one source. If you take an embryo, every cell in that growing embryo is capable of becoming any cell type. So you can see that depending on the conditions of the culture, will determine gene expression and make liver cells versus nerve cells versus blood cells. Now, generally, if you take an adult stem cell, so places that in the body that have stem cells would most commonly be someplace like the bone marrow. But if you take bone marrow, and apply those same conditions, you're really still only going to be able to make different types of blood cells. It's still a stem cell, but it's not a pluripotent stem cell because you can't take that bone marrow cell and turn it into, under regular conditions, just any other type of cell. However, we've figured out ways to be able to make cells induced pluripotent stem cells. So these iPS cells we can take cells and under a variety of conditions be able to coax those cells into becoming pluripotent. So taking a skin cell, for example, which doesn't necessarily even have to be doesn't have to be a stem cell to begin with, but we can kind of turn back the clock on that particular cell that was differentiated, get its DNA to express such that it now will be able to be coaxed into becoming any other type of cell within the body. So if a person, for example, has damaged heart tissue, and so they need some additional heart muscle to be transplanted into their body, we can't, normally the only recourse would be to actually transplant an entire new heart into that patient, which again has a low success rate. There's a high risk of infection. It's a very traumatic type of surgery. We need to have a donor, and there's not enough donors. They need to be able to match based on tissue types, and that's, again, it becomes complicated and has a higher risk of failure. Rather, if we can take skin cells from that patient, reprogram them using these techniques to become stem cells, those stem cells could then be induced to become heart cells, and those heart muscle cells could then be made into a tissue, and that tissue could be implanted back into the patient. This has been done for a number of simple tissues, so things like skin, things like the trachea, the esophagus, the bladder. It has not yet been done, and it's still many years away from being able to use this process to be able to grow organs, but that is definitely a field in which there's a lot of research being done right now. So perhaps in the future, this would be a way to be able to treat people with damaged organs or injuries that they suffer, is by reprogramming some cells within their body to become the part that needs to be replaced. So it's very exciting to be able to think about the possibilities there. Again, it's still quite a bit off into the future, but this overcomes some of the ethical issues that exist with having to rely upon embryonic stem cells for reasons I'll get into in a moment.